This is The Cell Cycle by Alex Woodworth and Terry Gibbs. The cell cycle consists of 4 distinct phases: G1 phase, S phase, synthesis, G2 phase, collectively known as interphase, and M phase, mitosis. M phase is itself composed of 2 tightly coupled processes: mitosis, in which the cell's chromosomes are divided by the 2 sister cells, and cytokinesis, in which the cell's cytoplasm divides in half, forming distinct cells. Activation of each phase is dependent on the proper progression and completion of the previous ones. Cells that have temporarily or reversibly stopped dividing are said to have entered a state of quiescence called G0 phase. After cell division, each of the daughter cells begin the interphase of a new cycle. Although the various stages of interphase are not usually morphologically distinguishable, each phase of the cell cycle has a distinct set of specialized biochemical processes that prepare the cell for initiation of cell division.
Geophase, the term post-mitonic, is sometimes used to refer to both quiescent and senescent cells. Non-pro-life cells and
Multicellular eukaryotes
generally enter the quiescent geo state from G1 and may remain quiescent for long periods of time, possibly identified as it often in case for neutrons. This is very common for cells that are fully differentiated. Cellular sentience occurs in response to DNA damage or degradation that would make a cell's progeny non-viable, and it is often a biochemical reaction. Division, such as a cell could, for example, become cancerous. Some cells enter the geophase semi-permanently, e.g. some liver and kidney cells.
Interphase. Before a cell can enter cell division, it needs to take in nutrients. All of the preparations are done during the interphase. Interphase proceeds in 3 stages: G1, S, and G2. Cell division operates in a cycle. Therefore, interphase is preceded by a previous cycle of mitosis and cytokinesis. Interphase is also known as preparatory phase. In this stage, nucleus and cytosol division does not occur. The cell prepares for division.
The first phase within interphase, from the end of the previous M phase until the beginning of the DNA synthesis, is called G1. G indicating gap. It is also called the growth phase. During the phase, the biosynthesis activities of the cell, which had been considerably slow down during M phase, resume at high rate. This phase is marked by the use of 20 amino acids to form millions of proteins, and later on enzymes that are required in S phase, mainly those needed for DNA replication. Duration of G1 is highly variable even among different cells of the same species. It is under the control of the p53 gene.
S phase. The ensuing S phase starts when DNA replication commences. When it is complete, all of the chromosomes have been replicated. Each chromosome has two sister chromatids. Thus, during the phase, the amount of DNA in the cell has effectively doubled. During the ploidy of the cell remains the same. During the phase, synthesis is completed as quickly as possible due to the exposed base pairs being sensitive to external factors such as any drugs taken or any mutagens, such as nicotine.
During the gap between DNA synthesis, G2 phase. During the gap between DNA synthesis and mitosis, the cell will continue to grow. The G2 checkpoint control mechanism ensures that everything is ready to enter the M, mitosis phase, and divides.
Mitosis. Mitosis is a process in which eukaryotic cells separate the chromosomes in its cell's nucleus into two identical sets of two nuclei. It is generally followed by immediate cytokinesis, which divides the nuclei, cytoplasm, organelles, and cell membrane into two cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the mitotic M phase of the cell cycle. The division of the mother cell into two daughter cells genetically identical to each other into their parent cell. This accounts for approximately 10% of the cell cycle. Mitosis occurs exclusively in eukaryotic cells, but occurs in different ways in different species. For example, animals undergo an open mitosis where the nuclear envelope breaks down before the chromosomes separate, while fungi such as yeast undergoes closed mitosis where chromosomes divide with an intact cell nucleus. Prokaryotic cells which lack a nucleus divide by a process called binary fission. The process of mitosis is complex and highly regulated. The sequence of events is divided into phases corresponding to the completion of one set of activities and the start of the next. These stages are prophase, prometaphase, metaphase, anaphase, and telophase. During the process of mitosis, the pairs of chromosomes condense and attach to fibers that pull as sister chromatids to the opposite sides of the cell. The cell divides in cytokinesis to produce two identical daughter cells. Because cytokinesis usually occurs in conjunction with mitosis, mitosis is often used interchangeably with M phase. However, there are many cells where mitosis and cytokinesis occur separately, forming single cells and multiple nuclei in a process called endoreplication. This occurs most notably among the fungi and slime molds, but is found in various groups. Even in animals in cytokinesis, mitosis may occur independently, for instance during certain stages of fruit fly and embryonic development. Errors in mitosis can either kill a cell through apoptosis or cause mutations that may lead to cancer. This was A Cell Cycle by Alex Woodworth and Terry Gibbs.